Welcome to this explainer on advanced thermoregulation. Today, we're unpacking the exact physiological mechanisms that keep mammals alive when the environment is basically trying to either freeze them solid or overheat their core. And we aren't just memorizing textbook facts today. If you're prepping for your A-level or advanced biology boards, you really need to know how to test these systems. So we'll be breaking down how these mechanisms actually look in lab experiments, from identifying independent variables all the way to analyzing physiological data. Let's get right into it. Okay, here is our roadmap for today. We'll start with the body's thermal baseline, move into the physics of heat exchange, map out the hypothalamic command center, break down thermoeffectors, and then wrap up with the clinical distinction between a fever and hyperthermia. Let's start with Section 1, the body's thermal baseline. So, 37 degrees Celsius. That represents the precise core temperature maintained by mammalian homeothermy. It's the specific, non-negotiable target our bodies fight fiercely to defend.
Right, and from a data collection standpoint, you have to remember that core temperature is the key dependent variable in thermal experiments. In a clinical lab setting, taking a tympanic or rectal reading provides a true core measurement. That's totally different from peripheral skin surface readings, which fluctuate wildly depending on the room temperature.
Now, why do we defend this specific number? Well, optimal enzyme activity occurs right at 37 degrees Celsius. High heat physically denatures proteins. It literally unravels their 3D structures and severely depresses neuronal function.
Exactly. Just picture running standard in vitro enzyme assays. If you set temperature as your independent variable and run trials at 20, 37, and 45 degrees, that 45-degree test tube won't just slow down. The reaction halts completely because the active sites are permanently destroyed.
So thermoregulation is really this constant minute-by-minute balancing act. Your body continuously weighs the heat produced internally by your organs and basal metabolism against the heat that's inevitably lost to the outside environment. And we can actually quantify this in the lab. If you put an animal model in a controlled metabolic chamber, you can measure their oxygen consumption to calculate heat production and simultaneously monitor the chamber's temperature changes to track environmental heat loss. It gives you the full picture. Moving on to Section 2, the mechanisms of heat exchange. Okay, here is your high-yield summary for the boards on heat transfer. Radiation accounts for about half of your heat loss via infrared rays. Convection is losing heat to moving air or water. Evaporation relies on the latent heat of vaporization, and conduction is heat transfer through direct physical contact. Now, if you were designing an experiment to test just one of these, you absolutely have to isolate your variables. To test convection, you'd place your subject in a wind tunnel to control airflow. To test conduction, you'd use a temperature-controlled water bath as a direct contact surface, while keeping the air completely still. Pay close attention to this next part, because it's a classic exam trap. Do not mix up conduction and convection. Conduction requires a stationary, direct physical surface contact.
Right, whereas convection requires a moving fluid. So, if an exam question asks about a subject lying perfectly still on an ice pack, the primary mechanism there is conduction. But if they're standing in a cold, flowing river, you are analyzing convection.
Section 3, the hypothalamic command center.
Deep in the anterior hypothalamus lies the preoptic area, or the POA. This acts as the brain's integration center, processing temperature readings from your skin. It then controls sympathetic output using EP3 expressing neurons and the inhibitory signal GABA.
And how do we actually know this? Classic lesion studies. If researchers create a localized lesion in the POA of an animal model, that animal completely loses the ability to thermoregulate in a hot environment. The POA is the undisputed control center.
When your blood temperatures dip below that 37 degrees set point, it triggers a negative feedback loop. The cooler blood washes over the hypothalamus, the POA detects the drop, and it immediately fires up the heat-promoting center.
It's a standard stimulus and response setup. The stimulus is the drop in the independent variable, the blood temperature. The response is the activation of the effectors. And because it's negative feedback, that response is actively designed to shut off the original stimulus.
All right, Section 4. Thermoeffectors, heating and cooling. First up, vascular control. Think of your cutaneous blood vessels kind of like the windows of a house. To cool down, vasodilation opens the windows, flushing the skin with warm blood to release heat. To stay warm, vasoconstriction shuts them, keeping all that thermal energy safely trapped in the core. You can literally visualize this data in the lab using infrared thermography. If you take an infrared image of a subject exposed to a cold room, which is your independent variable, you will see their extremities turn blue and purple on the scan, as vasoconstriction shunts the heat entirely to their core. Now, to actively generate warmth, your body turns on the furnace. As Aside from shivering, the major player here is brown adipose tissue, or BAT. By using a molecule called uncoupling protein 1, BAT actually short-circuits cellular respiration. It shunts proton fluxes directly into pure metabolic heat instead of making ATP. If you're analyzing lab data on this, look closely at the oxygen consumption. Because UCP1 uncouples the electron transport chain, those mitochondria have to burn through massive amounts of oxygen and lipids just to generate that heat. So a high oxygen uptake without any muscle contraction, that indicates high BAT activity. And for cooling down, we activate our internal AC unit through evaporation. When the sympathetic system triggers your sweat glands, your body heat is used to vaporize that fluid into a gas. This actively strips thermal energy away via the latent heat of vaporization.
Which is exactly why environmental humidity is such a crucial variable to control for in exercise physiology. If the relative humidity in your lab is close to 100%, the air is basically already saturated. The sweat secretes, sure, but that vital vaporization step fails, completely tanking your cooling efficiency.
Finally, Section 5, Fever versus Hyperthermia. Let's trace what happens during a fever. An infection introduces pyrogens. Your immune system then kicks off the arachidonic acid pathway to synthesize prostaglandin E2. PGE2 binds to EP3 receptors in the POA, dialing the set point upward from 37 degrees to 39 degrees. Basically, your brain now thinks you are freezing. In a pharmacology lab, this specific pathway is exactly what we target. If you administer a control group a placebo, they run a fever. But if the experimental group receives an NSAID like ibuprofen, it inhibits the cycle oxygenase enzymes, blocks PGE2 synthesis, and keeps the set point perfectly normal. Now, this is an absolute must-know exam trap. Fever and hyperthermia are not the same thing. In a fever, the thermostat setting in the brain is actively elevated. The body actually wants to be hotter.
Right. But in hyperthermia, like heat stroke, that biological thermostat is sitting completely normal at 37 degrees. The pathology there is that the physical cooling effectors, like sweating, have been totally overwhelmed by external heat. The body desperately wants to cool down, but functionally it just can't.
Hyperthermia progresses through stages of failure. It starts with heat cramps from fluid loss, moves to heat exhaustion, and finally hits heat stroke, where the core hits 41 degrees Celsius and cooling mechanisms shut down entirely. When you're analyzing clinical data for these conditions, look at the serum sodium levels and the presence of anhidrosis, which is the lack of sweating. A patient presenting with an extremely high core temperature but dry, hot skin, that indicates a fatal positive feedback loop of complete thermoregulatory failure. It really is a fascinating, deeply interconnected system. So, before we wrap up, we want to leave you with a provocative clinical question regarding this biological thermostat.
We know that a fever is a targeted, active immune defense designed to make the body hostile to pathogens. So if clinical trials show we constantly suppress every mild fever with antipyretics, are we actually skewing the variables to help the invading bacteria?
Think about that paradox as you review these mechanisms for your next exam. Keep studying hard, trust the structure, and we'll catch you on the next explainer.